Our third set of notes from the DNA unit is still covering Learning Target 1, identify the structure of DNA and explain how it can be copied, as well as serve as a code for making proteins or new traits. We're going to focus on this second part of how it can be copied and serve as a code for making proteins. So the main objective here is to describe the process of DNA replication. DNA replication, if you replicate something, you are making copies. So we're making copies of DNA. And one of the terms we can use, and we've had this term before, is synthesis. That means to make or to build. And so what we're doing is we're building or making new DNA molecules. We're making copies of the existing molecule so that we can have more copies of the DNA. And this process always occurs in the nucleus of the cell. So keep that in mind, that it always occurs in the nucleus of the cell. So the very first step that has to occur, remember, DNA is a double helix. It's made of two strands of those nucleotides joined together. And they're joined together by hydrogen bonds between the base pairs, C with G, A with T. Well, the two sides of the DNA molecule are unwound and separated by an enzyme called helicase. And what that enzyme does is it breaks the hydrogen bonds. Remember, we said that hydrogen bonds are very weak bonds. Well, I told you this would be important, and the reason it's important is that allows the helicase to break those weak bonds and separate the DNA into two separate strands. Okay, because the only thing joining them together are those hydrogen bonds between the bases. Each side then acts as a pattern or a template where a new strand can be copied off of. So when we separate that DNA into its two separate strands, each strand acts as a template or a pattern to be copied off of to make a new DNA molecule. There are also special proteins that kind of hold those, because DNA, because it's a double helix, likes to keep winding up. There are special proteins that kind of keep the DNA strands separated. Once that helicase has broken those bonds, it keeps them separated into two strands. So you can see here the picture. You have your original DNA, and now the helicase came through and it broke the bonds and separated it into two separate strands. And here it's showing you an example of the helicase there. The next thing that happens is another enzyme called DNA polymerase moves down the exposed strands and puts down new nucleotides. These are just kind of floating around in the nucleus, and it starts putting down those new nucleotides in a specific order according to the correct base pairing. Remember, I said you would have to remember how these bases pair up, A with T and C with G, because now DNA polymerase has to put down the correct base, the correct nucleotide, according to which base should be paired up with the open strand. So here we have the closed strand. It's starting to open up there, and now we have these nucleotides out here that are floating around, and DNA polymerase will start matching up. A with T, and now this G is going to go here with the C, and it's just going to keep, one DNA polymerase will move up one side, another DNA polymerase will move down the other side. And you can see up here, here's the DNA polymerase moving down the template strand, and it's adding a new strand right in there. Each new DNA molecule will be half new, that's been newly copied, and half of the old or original strand. This is called semi-conservative replication. You need to know that term. Semi means partially, so it's partially conserving a part. That's because it always conserves or keeps half of the original strand of DNA. So here, if we had this DNA strand here, it's separating. What separates it again? Helicase. Separates them by breaking the bonds. Here you have one strand. Here you have the other side of the original strand. And then you have free floating nucleotides. DNA polymerase is going to put down each of those in the correct order. and match them up according to how they pair up. And in the end, what you end up with is 2 identical strands of DNA, just like you have over here. Half new, half from the original. That equals semi-conservative. Okay? Now, why is this process so important? Every single time your cells divide, your DNA has to get copied. Why is that? Well, remember, your DNA holds the code for telling the cell how it's going to function. If the new cell never gets any DNA, it won't even be able to function and it will die. So it's very, very important that every single time your cell is going to divide, DNA gets copied. This occurs during what we call the synthesis phase in interphase. And we'll be talking more about replication and cell division in a later unit when we start the second semester, but we'll just kind of introduce that here. Again, remember that DNA replication occurs in the nucleus, and there's lots of replication sites. This is showing this splitting open right here, and you have the original strand there, the blue one, and then the newly copied strands, the gold along the side. But this would also be happening up here, all along. We used to always compare DNA replication kind of like to a zipper, like the helicase would break the bonds. It'd almost be like if you're pulling down the zipper on your jacket or something, it goes from one end to the other. Well, it's more like, have you ever had with your jacket where your zipper is kind of broken and it opens up at different spots along the way, and you have these little gaps in your zipper? Yeah, that's more like what DNA replication is that you have helicase moving at different spots there, opening up these places so that different parts of the DNA are replicated at different times, and that makes it a lot more efficient. So lots and lots of replication sites so that you can have faster copying of the DNA. Now, if you're going through something quickly and writing something down, you might sometimes have a mistake or write something down incorrectly or put the wrong letter in or the wrong word in. Have you ever done that when you've texted? Yeah, and autocorrect tries to correct it and makes it even worse sometimes. Well, your DNA gets proofread. There's another type of DNA polymerase that actually goes along and proofreads and makes sure that if a C got bonded, with an A instead of with a G, that it would take that and cut that out of there and put the correct base in. Okay? So DNA gets proofread, so it minimizes the errors. Doesn't mean there aren't any mistakes made. About 1 error per billion nucleotides, that's pretty good. So there are still some mistakes made now and then, but overall, DNA replication is a pretty accurate process. And there you have DNA polymerase going down, putting down the new nucleotides as it goes along. DNA polymerase is going to proofread as well and double check things. And so there you have the helicase unwinding the 2 DNA strands as you go along as well. So just to practice a little bit and looking at some of these different parts, let's see if we can label some of the structures that the arrows are pointing to. So helicase, remember the job of helicase is to unwind and separate the 2 strands. And so this right here would be your helicase, this structure right here. The free-floating nucleotides, well, we have some right here. There's also some down here as well. And those are your free-floating nucleotides. That is what the DNA polymerase right here, here's some more right there, DNA polymerase, that's this molecule right here, is going to take those and match them up according to the correct base pairing. And it's going to keep moving on down the line, putting down those correct nucleotides as it keeps going along. And then this right here, the outer part here on both of these, this is the old or original DNA strand. I like original rather than old because you know you guys, I'm pretty old myself, so so I don't necessarily care for that word. So we'll just say original DNA strand. And then you have the newly copied right here. This is the new DNA strand. Okay. So DNA replication is pretty straightforward. Again, it happens in the nucleus because your DNA, the reason for that is your DNA can never leave the nucleus. Your DNA is so important, it stays there always because it contains your coded message for how your cells are supposed to function, for how what proteins need to be made in your cells, and so on. So DNA replication occurs in the nucleus. It has to happen every single time your cell's going to get ready to divide, and it helps make more DNA so that the new cells can have the coded instructions that they need in order to function and make the proteins that they need.